All right, good afternoon everyone. It's a great honor and pleasure to me to introduce to you today Professor Bonn from the Bonn Institute in Amsterdam. Well, the title of the talk you can read for yourself. I'm just going to tell you in brief a little bit about Misha's background as he told it to me. He took his PhD in 1996 at the Bonn Institute actually. And then he made for a postdoc at the Fritz Haber Institute, working with Martin Wolf and Gerhard Adel, stuff like surface science, right?
Yes.
Then also at the same time, during the 3-year postdoc, he worked with Tony Hines at Columbia University. After that, he went back to Holland, to Leiden University, and was there working as an assistant professor, and after that also associate professor. Finally, he got then a position as a group leader at AMO, and the topic there is biosurface spectroscopy. And, well, I don't have much more on the list to read here, so I will just leave the word to Misha and ask you to thank you for coming here.
Well, thanks for inviting me, and thank you all for being here. So, I'm going to discuss our work on water at biological membranes and how we've been trying to understand the structure of that water, and that we can learn something about the structure through the dynamics, and how we can use that water for biomolecular sensing. And it's obvious from the next picture that my group really likes water. So here we are on our group outing. It was a very, very nice day. Unusually nice for Holland, in fact. So a lot of the people on this photograph, I don't think it's much use if I tell you their names, because you'll probably forget them, but a lot of work was, of course, done by them. We're in Holland, we have a queen, and when she talks about herself, she just does that in plural, and it's the majestic plural, and it means, when the queen says we, the Queen of Holland, it means her and basically all of Holland. And I think there's also such a thing as the supervisor plural. So when I say that we have done a lot of work on, understanding water at biological interfaces, it actually doesn't include the supervisor. It actually means that these people have done a lot of work to understand water. So it's sort of the opposite of the majestic plural. So anyway, we've done a lot of work on interfacial water. The outline of my talk is very simple. I'll introduce sort of the questions that we have, tell you briefly about some of the qualitative structural things and how we can use that for sensing. Then I'll tell you that what was thought we knew about structure is in fact not true. Things are a little different, but that we can learn a lot about the structure from the dynamics, and then I'll conclude. So feel free to interrupt me. The talk is actually, I don't think, that long. So if you have any questions, just yell. So this is a cell membrane. And the basic building block of the cell membrane are these phospholipids. They're little balls, shown enlarged here, that are hydrophilic polar head groups. They're usually charged. And the two sort of wiggly lines are apolar hydrophobic alkyl chains. And when it put in contact with water, these phospholipids self-assemble into such a bilayer. And this is the box around a cell, right? This is what separates the inside of the cell from the outside of the cell. Now it's important. It's an important box because it controls what goes in and out of the cell, and a lot of important biomolecular interactions occur at the cell membrane. And for a long time, it was thought that what the cell membrane is just sort of a static, it's sort of a barrier that separates the inside from the outside of the cell. And in this barrier, there are the things that give this barrier its function, the membrane proteins, the transmembrane proteins. And these proteins determine what can go in and out of the cell. Now, this picture we now know is not correct. This is a very crude oversimplification. the functionality of the cell membrane is really determined by a very subtle interplay between the lipids, the proteins, and the water. And to tell you why this interplay is important, it's well known that some of these transmembrane proteins don't function in a cell unless specific lipids are present. So they require specific lipids to work. The reason why they then don't work has been thought to relate to the nature of the water-lipid interaction. So it's the hydrogen bonding nature of the lipid head groups that if you change that, then the protein functionality changes. So it's clear that the water, the lipids, and the protein sort of act together to allow the membrane to do what it does. Now, in the talk that I give now, I will focus primarily on water. We've done work on lipids previously, and actually Sylvia Roop, who's in the audience, did a lot of that work. But I'll focus today on sort of one of these three main building blocks, namely water. Now, the things that are known from membrane-bound water has been mainly obtained from MD simulations, and I have an example here. So you see here a bilayer. So this is the center of the bilayer. So these yellow things are the methyl groups of the lipids, and you can see there's some sort of very clear qualitative things you can say when you look at this simulation. The water that's penetrating into the membrane is sort of fixed. It's not very mobile, whereas the water that's further away is much more mobile, and it rotates and it moves and it diffuses much faster. So there's clearly such a thing as membrane-bound water, right? Some specific interaction. Now there's some questions about the residence time, about the preferential orientation of water molecules, how heterogeneous is the water. And it's very hard to address these questions from an experimental point of view. And the challenge is that you want to look at this sort of one molecular layer of water, these few molecules at the membrane interface, without looking at the bulk water that's underneath. So the challenge is to directly probe membrane-bound water. Now, what we do in our sort of physics institute, we're not a biology institute, is we use model systems for membranes. And in fact, such a self-assembled monolayer of lipids on water is a good model for a cell membrane, because, of course, in reality, a real membrane is a bilayer, so it's this system mirrored in this plane. But it's known that the two leaflets, as they're called, of a cell membrane are very weakly coupled. So the fact that there is no membrane here doesn't affect the water that we're interested here. Now the second thing that we always do is we always compare the water-lipid interface with the water-air interface, because there's kind of trivial effect that when you sort of end the water structure here, you interrupt the hydrogen bonded structure that makes water so unique. So we want to be able to distinguish sort of trivial effects due to the fact that we sort of cut open the water surface, and we want to look at the specific interactions that the water has with the lipids. So that's the reason for comparing these two systems. Now, the way we look at the water is using vibrational spectroscopy, because the vibrations of water are very good reporters of their local surroundings. And in fact, if you look at in bulk water at the hydrogen bonding, which of course is what makes water unique, then you can see that if the hydrogen bond is strong, so it means small OO distance, then the OH stretch vibration, so that's this motion of the water molecule, the OH stretch vibration has a low frequency. And if this hydrogen bond is weak, it has a high frequency. That's this plot here. So we can use these water vibrations to tell us something about the local environment of water without the need of, say, fluorescent probes. But then the question is, how do we look at this one molecular layer? Well, for that we can use a type of spectroscopy that's called some frequency generation, which provides the vibrational spectrum of a monolayer of water, which is precisely what we want. And in these experiments, we sort of excite the water vibration with an infrared laser pulse, and then we use a visible pulse to upconvert that infrared, and we generate the sum frequency of those two laser fields. I don't want to discuss the details of the technique. There's two things that are important. This will give us the vibrational spectrum of these interfacial water molecules, and the nice thing is that it will only give us the vibrational spectrum of interfacial water molecules, because this mechanism is forbidden in bulk media. So as soon as we're a few water molecules deep, this process can no longer occur. So it's a surface-specific process. And to illustrate the surface specificity, I show you here the SFG spectrum here in light blue of the water-air interface. And you can see that it's very different from the bulk vibrational spectrum of water shown here in gray. And most notably, you see that there are sort of three peaks in this interfacial water spectrum. There are two peaks sort of redshifted in the hydrogen bonded region, and these have been ascribed to strong hydrogen bonded in like ice-like domains at the surface, and sort of weakly hydrogen bonded in water-like domains at the interface. And this very high frequency peak here at 3700 wave numbers is due to the OH group that sticks out of the surface. So it's not hydrogen bonded, and therefore has a very high vibrational frequency. And of course this is, well, the fact that there are three peaks are very typical for the surface of water. So how can we use SFG to learn something about the structure of water? So in what I'll show you in the following, I will spend a lot of time discussing D2O, so heavy water, but as you can see it shows the same type of response that we get for H2O, and the reason why we use D2O is that the frequencies are a little lower and that's a little more easily accessible with our laser system, but it doesn't change the story. Now if we put a lipid, a monolayer of lipid on the water molecules, you can see that the spectrum changes dramatically. There's actually two changes compared to the water-air interface. The first is that the water signal shoots up by a factor of 20 or so, and the second is that we get another signal that we can relate to the CH3 vibrations of the lipid. Now this second observation is not so strange, because what's happened is that going from one interface, the water-air interface, we now effectively have two interfaces. We have the water-lipid interface and then the lipid-air interface. So we get two signals because we have two interfaces. And this signal here comes from the lipid-air interface. But the surprising thing is that the signal from the water-lipid interface is so much higher than that from the water-air interface. Now, one of the things that might contribute to that is the fact that these lipids are generally charged. And they can have a net neutral charge, they can be Twitter-ionic, but in general they have charge. So this is the structure. There are phosphate groups, there are ammonia-like groups and carboxylate groups. And maybe that has something to do with the fact that the signal of the water is so large. And indeed, if we use a cationic lipid, so it's just one positive charge, we also get this huge enhancement. So that implies that the large signal is related to charge, and this actually kind of makes sense. Because if we look at the water-air interface, you expect this interface to be pretty disordered. So the symmetry is broken already very quickly as you go into the bulk. And that means that we're not sensitive. With our technique, we're sensitive to broken symmetry at the interface. That means the signal will be relatively small. So if we now put a layer of positive charges, those lipids, on the interface, the dipoles of the water will reorient in such a way, that I've sketched as a cartoon here, that will break the symmetry sort of going deeper into the bulk. And that means that SFG is now allowed sort of a few molecular layers deep, which will increase the signal. Now indeed, if we add sodium chloride, sort of tens to hundreds of millimolars to the solution, you can see that the signal of the water decreases continuously as we increase the salt concentration, and that we can then understand by noting that the chloride ions will sort of absorb onto the positive ionic layer and will reconstitute the disorder, restore the disorder in our layer. And we can sort of quantitatively understand what's going on by relating the water signal that we observe to the surface potential through the Gouy-Chapman model, where the Gouy-Chapman model states that the surface potential is a function of surface charge density and electrolyte concentration. And you can see that this simple Gouy-Chapman model describes our data quite nicely. Now, this is a sort of nice observation, but it's not particularly useful, I think, to the world. What was useful was the idea that we had that maybe we could use this for detecting DNA. So, biomolecular sensing of DNA. Now, the reason why we thought that was interesting was that there's a lot of interest in detecting DNA, and the way this is generally done is that people use fluorescent labels, attach the fluorescent label to the DNA, add the complementary strand, immobilize the complementary strand on a glass plate, and as soon as the DNA binds, you can tell with the fluorescence microscope that you have the complementary DNA strand. Now that's, of course, a very sensitive technique, but it's an expensive technique because you have to do this labeling very efficiently, and it's not very easy. So there's been a lot of interest in label-free DNA detection schemes, and the new schemes that have been developed over the past few years, I've listed a few here, are all based on sort of lithography with varying degrees of success in terms of the sensitivity of detection, but they all have one thing in common, which is that the detection is electronic rather than using a fluorescent probe. And the sort of basic idea behind all these detection schemes is that you have some sort of transistor type device, and DNA is a polyanion, and as the DNA absorbs onto the surface of the silicon transistor, the electrical properties of that transistor change because of the field that's exerted by the negative sort of monolayer of DNA, and therefore you can detect the presence of DNA by a change in the current. Now, it's very elegant. You look at me as if I'm talking gibberish. Should I try to explain it a little more? Okay, so let's just say that the essence is that you can electronically detect a layer of negative charge when the DNA absorbs onto this sort of silicon device. Now, it's very nice, but you still have to make a lot of effort. So you don't have to work on the DNA, but you have to do some sort of nanolithography and biochemistry, in fact, to make this work. And so you're sort of moving the problem around, was our feeling. So our idea was, well, we're very sensitive to ions through the water, and why don't we detect optically and not electrically, using water as a reporter for the presence of DNA. In the same way that we could detect sodium chloride, of course with like sort of millimolar sensitivity. So here's again the SOG spectrum of water underneath the lipid monolayer with no DNA added. And then indeed as we add DNA, you can see that again the water signal goes down. But the nice thing was that it already goes down in the picomolar range. So we have a very sensitive way of detecting DNA in a relatively simple way using water as a reporter for the presence of DNA. Now, of course, this example is non-specific, but it's very easy to make this sensitive to a specific sequence of DNA. Now we can also understand why the sensitivity is so high by looking at a sort of modified Gooey-Chapman model, where we have to, so this is why this concentration scale was so long. So here are the points for DNA. And now we have to modify the Gooey-Chapman model, so this is the equation I showed you before. So the surface potential is a function of the surface charge, and of course the fraction of available charges at the surface. So that means charges where no DNA is yet absorbed, and the near surface concentration of DNA. The near surface concentration of DNA is determined by this Boltzmann factor. So there's on the one hand an attraction, electrostatic attraction of DNA towards the interface, but there's an entropic force that wants to keep it into the bulk. And then there's sort of a modified Langmuir adsorption behavior, where if you have a lot of empty sites, the probability of DNA adsorbing is large. And now the thing that gives us this very high sensitivity is this very small thing here, n. This is cooperativity factor. And the reason why we have some cooperative adsorption of DNA is that if you have, in this DNA strand, if you have one negatively charged base pair adsorbing to the membrane, then there's necessarily a second and a third negatively charged base pair very close by that has a much larger probability than statistically to adsorb to the interface. And this is this so the adsorption in a way is cooperative. That's what we mean here. And indeed we can describe the data very nicely with a single base adsorption coefficient that we can determine independently, and then a cooperativity constant of 3. So that's so the nice thing is that we now have a label-free way of detecting DNA, which doesn't require any lithography. The problem is that it requires an amplified infrared laser system, which is very expensive, but we're trying to think of ways of circumventing that. So this is very nice, and I've shown you that we can use this large signal for sort of useful purposes, but of course what we're interested in is what determines the shape and also the amplitude of this signal. And what does this tell us about the structure of the water? Now, and you can see that there's 2 question marks here, because it actually doesn't tell us very much, and I'll explain to you in the next few minutes why not. So here I show you the 2 spectra of, this is H2O once more, in the hydrogen bonded region of the water-air interface and the water-lipid interface. And you can see that there are subtle differences, but sort of, if you squint your eyelids, they're pretty much the same, right? There's sort of 2 peaks, 3,400, 3,200. The ratios vary a little bit. And in fact, for every water interface, you always see these 2 peaks. And they vary in amplitude, and there's subtle variations, but you always see these 2 peaks. And it was not clear to us why that was. And so I briefly told you that the presence of those 2 peaks for the water-air interface was explained as being due to strongly hydrogen-bonded interfacial water molecules in an ice-like environment, and weak hydrogen-bonded water molecules in a water-like environment. Now, this interpretation implies a couple of things. For one, it implies that the surface would be more ordered than the bulk, that there would be, ice-like structures at the interface. I've been a surface scientist for a number of years now, and I don't know of any system where the surface is more ordered than the bulk. You have things as surface melting, but you don't have things as like surface solidification. The interface generally is more disordered. The second thing is that we know that from bulk measurements that water is very, very highly dynamic. These hydrogen bonds rearrange very quickly, so that also makes it sort of implausible that there are really domains of ice-like and water-like structures. The interconversion should be much faster than what's possible to show up in this spectrum. So is this hypothesis correct? And this is like a 15-year-old hypothesis. This paper has been cited over, well, 200 times or something. And, well, the first sort of stupid thing that I thought was, why, you know, this is the water molecule. If it's D2O, you have two OD stretch vibrations, and these are known. This is from the gas phase. They're known to split up into a symmetric and an asymmetric mode. You know, we see two peaks. Why are those not simply the symmetric and the asymmetric mode of water? So then, now we have two hypotheses, right? We have the strong and weakly hydrogen bonded hypothesis, and we have the symmetric asymmetric stretching hypothesis. And I'll show you that in fact they're both wrong. But before I show that, I'll show you how we tested the hypothesis, which was by isotopically diluting water. So we started looking at HDO, where we replaced one hydrogen by deuterium. And when you do that, if there's like ice-like domains and water-like domains, you will still have, in our case, OD or OH groups that are strongly and weakly hydrogen bonded. So the double peak structure will persist, even when you isotopically dilute the water. If you have a symmetric, asymmetric, if that hypothesis is true, then in fact the two peaks will collapse into one peak, because you've now broken the symmetry within the molecule, and you can't have this splitting anymore. So if this is true, you keep the two peaks. If this is true, you would expect the two peaks to merge into one. I hope this is clear. If it's not, now is the time to interrupt. So when you do the experiment, you see that in fact these two peaks will clearly collapse into one. So this is for pure D2O, this is for HOD. Same thing for the water-lipid interface, you end up with one peak. So you would think that this symmetric-asymmetric stretch interpretation is correct. Well, at least it's clear that the interpretation in terms of substructures is incorrect, that we know. Now, if we now look at the frequencies that we find, then they show that this sort of symmetric-asymmetric stretch story is incorrect. And I'll show you why. So this was the hypothesis that we had. Now, if we then go to isolated HOD, so if we switch off this coupling, you would expect that the OD stretch of this molecule would be right in between those two, right? Well, that's actually not the case. It may seem like that, but the resonance is actually, there's some interference that we can account for, but the resonant frequency is actually slightly higher than the peak of what we thought was the asymmetric stretch. And this then is totally inconsistent. I mean, there's no way that you can explain that this guy would suddenly jump up here. This is just not consistent with the symmetric-asymmetric stretch. And in fact, what we saw was that these two bands have the same symmetry, which is also inconsistent with asymmetric-symmetric, because you would expect to have different symmetry. And so what actually happens is that in addition to the stretching mode, there's this, I've tried to do it in PowerPoint here, but it's not so easy. You have the bending mode of water, which is this mode here. And the overtone of this bending mode has the same frequency as the symmetric stretch. And so it interacts with the symmetric stretch. And it's in the same way that these two split, it now splits into two new peaks, and those are actually the ones that we observe. And that explains why these two peaks have the same symmetry, and it also explains why the isolated OD has a higher frequency than the two peaks that we observe. So what this tells us is that, in fact, the spectra that I showed you, they don't contain information about the structure of water. They contain information about some intramolecular vibrational coupling effect of water that is really not maybe all that informative. These results are interesting from a different point of view because they show that these ice-like and water-like substructures are not present at the interface, and that in fact the water interface is structurally much more homogeneous than we thought so far. So the problem that these sort of broad peaks don't tell you very much is the same problem that people have encountered in bulk water. And there's a sort of trick that people have used for a decade or so, and that's looking at the dynamics of the vibrations. And so we did the same thing. So we have our normal SOG setup, but now we simply add, this is a typical example of a supervisor plural. We simply added a pulse. I assure you that this is a lot of work, and I didn't do any of it. So we added this infrared pump pulse, and what that does is it excites a significant fraction of the water molecules to their first vibrationally excited state. So it depletes the ground state, and that means that less water molecules are available for this SOG process, so it means that the SOG intensity will decrease. And as soon as, if we wait long enough, the population will decay back to the ground state, and the SOG signal will recover. So what we expect to see in such an experiment, if we vary the waiting time between this pump pulse and this SOG pro pair, is that the SOG intensity goes down, and then the recovery will tell us how long this vibration lives. And I will show you that this vibrational lifetime contains important information about the structure of membrane-bound water. So we did these experiments at 4 different frequencies within both of these bands. And for the water-air interface, we get typically these kinds of traces. So you see at 3500 you really get this nice decrease in SOG intensity and at least a partial recovery. At 3200 you get a decrease and then an additional decrease. And this is due to the fact that when we excite the water molecules and when they relax, the system heats up by about 10 degrees centigrade. And so there's a sort of a thermal effect in here which shifts the long time signal. Now if we did many more experiments like this, and there were a few sort of remarkable observations that there's only 2 time constants in there. There's sort of a fast time constant, which you see everywhere here, and then there's sort of the slower time constants, which you see here. And the second remarkable thing was that the signal did not depend on the polarization of our pulses. So we do laser spectroscopy, and we always find that if you excite along with your polarization along one axis, then you excite the water molecules that have OH groups aligned along that axis. Then if you probe along that axis, you see a large signal. But if you probe perpendicular to that axis, you see a much smaller signal. And this is what you always get, but not here. There was no dependence on the pump polarization. That was very surprising. Now, in fact, all these observations have also been made, are reminiscent of bulk measurements. And the explanation is that after you excite a water molecule, or a fraction of water molecules, there's very fast and very efficient energy transfer, vibrational energy transfer between water molecules. And that explains why the orientation that you impose by exciting along a specific polarization axis is lost, because as soon as this takes place, the transition dipole moment, of course, can effectively rotate. It doesn't require rotation of the molecule, it just requires hopping of the excitation. Then the molecule relaxes, and during this relaxation process it becomes sort of internally hot, and then it takes a certain equilibration time for the energy to be equilibrated to give rise to this temperature increase. And in fact, what that means is that we can excite surface molecules but the excitation sort of runs around and goes into the bulk and comes back to the surface, and there's this very rapid exchange of vibrational energy between surface and bulk molecules. And it also means that the excitation, if we excite, say, very strongly hydrogen bonded molecules, the excitation will sample all possible water configurations. So we don't expect to see a dependence of the lifetime on the frequency. That's precisely what we observe. So we can indeed model, those are the lines that you see here, we can model our data with, these are in fact lifetimes obtained from bulk measurements, and that means that there's a negative way of saying that, which is that we did a very, very difficult experiment to show that the people who did these measurements previously in bulk did them right. There's also a positive way of approaching this, which is saying that, wow, look, there's this very fast and efficient energy transfer between interfacial water molecules and bulk water molecules. We prefer the latter approach. In any case, the question was, well, these spectra are very similar, so you might expect that the dynamics are also very similar. And that's in fact not true. So here you see a comparison of water at the water-lipid interface, and you can see that the dynamics are very different. And in fact, the model that you see here is simply an exponential recovery of the signal with a characteristic lifetime. We find no evidence for this very fast spectral diffusion, so no evidence for this energy transfer. We also do observe a polarization dependence here, which also tells us that this excitation is not hopping around. And so the picture that emerges is that we excite sort of interfacial water molecules here, and the excitation remains at these water molecules until they relax. And in fact, we can model the dependency of the lifetime on frequency by a model of isolated hydrogen-bonded water molecules very nicely. Now that's a kind of a puzzle. So what we see is that at the water-air interface, there's this first energy transfer between the interface and the bulk that's very efficient. I mean, it's faster than 50 femtoseconds at least. Whereas for the water-lipid interface, we find sort of isolated water molecules that exchange energy with the bulk on timescales longer than picoseconds. Now, it actually sort of took us a year to figure out how that was possible. And the reason why it took us so long was that the picture that we and many other people have of a lipid interface is that it's a sort of atomically flat interface of head groups that are sitting on the water, and then there's this sort of bulk water underneath. And that picture is simply not true. And the answer to this puzzle lies in the steady state SOG spectra. So one of the things we can do with SOG is we can determine molecular orientation. And I don't want to go into too many details. You can ask me later if you're interested. But when we determine the orientation of the water molecules underneath a negatively charged lipid monolayer, we find that they're sticking with their oxygen molecules towards the negative head group. And this puzzle is even more. So it doesn't make any sense. Why would the electronegative water be sort of sticking towards the negative head group? Now, the answer to both these puzzles is that we're actually looking at water that's above the head group, that's more inside the membrane, which is actually what I showed you was apparent from these MD simulations in the beginning. These, of course, will be oriented in the way that we see them to be oriented, and also they are spatially isolated from the bulk that will be sort of underneath here, and it explains why the energy transfer is slowed down. So we can learn something about the structure from the dynamics. That's the message of the last part of my talk. So with that, I'd like to conclude. And I've shown you, hopefully, that the structure of the water surface for any water interface is simpler than has been thought. So this ice-like, liquid-like is just not true. It behaves actually much like just a liquid water. At every interface that we and other groups have studied, we see this very fast vibrational energy exchange of the interfacial water with the bulk. The only interface where we don't see this is for the membrane. And this proves that the water there, this membrane-bound water, doesn't just terminate the bulk. And that's sort of my take-home message here. A membrane is a collection of lipids and, of course, proteins and water. The water is really an integral part of the membrane. And we can use this water signal for sensitive, label-free DNA detection, as I've shown you. And with that, I'd like to thank you for your attention.
So, thank you very much, Misha. So, do we have any questions right now? Yes, please.
You show the spectrum of water, and I basically have two questions. You have at the beginning of your talk, you have an increase into the bonded OH when you target the membrane on top of the water, and you attribute that to charges of the head groups.
Right.
Did you count that this OH may bond actually to the head group and may increase the number of bonds, not only because of the charges, but you get an increase into the signal?
Yes.
Because of this is bonding to the ethylene groups in the head group.
Yes, I think that's a very good point. So that's basically summarized in this picture here. So it's not only the alignment of water molecules, but it is indeed also the increased number of water molecules that contribute to the signal. I think it's a combination of both of those things. And yeah, so one of the things we're now trying to do is try to figure out where exactly these water molecules are. Because of course it's a very heterogeneous environment for these water molecules. And one of the things we're trying to do is do a type of two-dimensional spectroscopy where we excite specific vibrations of the lipids and look at the effect of that excitation on the water molecules, a bit like 2D NMR.
Because basically that head loop is red. I mean, it's like oil. It's not stiff.
Yes, no, absolutely.
So
in fact, this angle is closer to 90 degrees than to 180 degrees, as I've pointed here.
And then the second is you have on the spectrum of water, you have a little bump at around 38 or a little before. Did you assign that to something, or?
That might be just noise, but I'll...
Oh, yeah, that one.
You mean this guy here?
Yeah.
Yeah, I think that's just noise.
Just noise.
Can you go to this slide where you had the interference between the overtone of the bending mode and the stretching mode to show something? Did you try to do experiments the water air interface in the fundamental bending region?
No, that would be very nice.
Yeah, so I was thinking, because as far as I know, no one has detected the bending mode at 1500 or 1600 with
Right.
water. So why do you think the bending mode would influence the spectrum in the overtone so much if we can't detect the fundamental bending mode itself?
Yeah, so it's not the fact that it's optically inactive doesn't mean that it can't couple to optically active modes. I think that's one part of the answer to your question. The second part is that I didn't think of this interpretation. This is an old interpretation. This coupling is sort of known in ice, in water ice. There you see it. I think you don't see it in water, liquid water, because there's too much inhomogeneous broadening, so it's just smeared out. And it's also been used in the interpretation of Raman spectra of bulk water, but there it's very controversial. And it's harder to make these kind of sort of harsh statements that we can make with SNG. Does that answer your question?
Yes. And also another one. You said that this strong, weak hydrogen bonding you didn't believe in.
Right.
So do you think all hydrogen bonds have the same strength? Or
No. Actually, what you can see, I don't have a picture. So what you really see is that the ratio of these two peaks really varies as you change the type of lipid. And the reason for that is that if so I've drawn this as one distinct frequency, but this is a sort of inhomogeneous band. There's a lot of different frequencies for the symmetric stretch. And what happens is that the overtone of the band doesn't shift. So if there's a continuum of states here that's shifting as you vary the hydrogen bonding strength, but the whole stays at the same position, then what you get is variations in the relative intensities of those peaks. And that's, for instance, also what you see when you vary the temperature. So this interpretation actually explains a lot of observations. So variation of temperature, variation of pH, variation of salt concentration. And so the experiment to do, if you want to determine the hydrogen bond strength, is to measure the frequency, the central frequency of the isolated HOD mode, the uncoupled mode. And that actually varies significantly with the type of interface.
As a comment on that, with X-ray emission spectroscopy, we actually see two specific components. One is very disordered, and the other is very ice-like or tetrahedrally coordinated. And then we asked the question whether these two components are spatially separated or not. And with small angle X-ray scattering, you actually see that there are structures of the order of 10, 20 angstrom in the liquid. So, I mean, they may not exist at the interface still, but that seems to be the structure of bulk,
Right.
at least. I had a question, which was, okay, actually two questions. I mean, when you say you detect DNA, I mean, you're really detecting the charges.
Yes. Yeah, so a very simple way of making it sequence specific, which is what you would like to do, is to, and still label free, is to insert neutral DNA, PNA, into the monolayer. And that will bind the DNA, and the DNA will then still absorb, will specifically absorb onto the interface.
But how do you discriminate between DNA and ions?
This is a very long discussion. So one of the key parameters is the ionic strength. So if you have single ions, we need to put in hundreds of millimolars or tens of millimolars to get the same effect that we get with picomolar DNA concentration. That's because the ionic strength of DNA is much larger. So regular ions won't affect the signal unless they're at very high concentrations. And then you can make it specific by inserting PNA into your monolayer. That's a common approach.
Yeah.
If I may chip in on the last question. If it's so that charge specific, could you imagine that proteins would give the same response?
Yes.
So how would you distinguish between DNA and proteins?
So again, we've actually used this observation not only for looking at DNA, but we are actually also looking at protein and peptide membrane interactions. So when we put a protein in the subphase, depending on the specific lipids that are present, it can bind to the membrane, and we can detect that binding both through vibrations of the peptide but also through the water. So yes, if you want to detect DNA, you're going to have to know what the DNA is in, what other things are there, and well, the simplest trick is to use just a very, to use not a lipid, but a very simple, sort of chemically inert cationic surfactant. That will just do the trick.
Can you comment a little bit more whether your model of this intramolecular coupling applies also to bottom water?
Well, yes and no. So you can see that the bulk spectrum has some structure. So if you freeze it and it becomes ice, really ice-like, then you also get this, it's called an Evans window, so this dip in the middle. I think bulk liquid water is a very difficult substance, and I don't know to what extent this Fermi coupling here plays a role. It's clear, it seems evident that for ice it does play a role, and I can't comment on what happens in water.
Is there a reason that it should not take place in white water?
Well, one reason that it might not show up so clearly in the IR spectrum is that, well, you expect there to be a lot of inhomogeneity in the system, as was mentioned already. But so in the Raman spectrum, it seems to be there. You get these two peaks, and the window is right at the overtone of the band.
Okay, there was one more.
So again on this Bonn region, why wouldn't be these two peaks, this Fermi coupling, like in the CH region where you have a Fermi resonance, where it's a well-defined peak? Maybe you said this, but I didn't get it. Why don't you see sharp peaks and well-defined peaks? Why it's such a broad
Yes.
line shape?
Well, I think that's just inherent to water. So that's one of my very first slides. So there's just this very strong dependence of, which we can use to learn something about water, of the OH stretch vibration on the hydrogen bonding strength. And there's just a lot of variation in the hydrogen bond strength in, I think, at least interfacial water. And so that makes everything very broad. But that's a whole other discussion that the interfacial water community also has. What is the degree of inhomogeneity of this interfacial water? And I think our results show that it's not as inhomogeneous as was thought initially, but I can't, right now, with our techniques, I can't comment on how homogeneous it really is.
All right, so time is running. Oh, perhaps we should leave the final question to Sylvia then?
One
No.
comment. So basically what you're saying is we have this Fermi resonance splitting,
Right.
but we still have these strong and these weak hydrogen bonds.
Yes, so
basically in the whole argumentation you insert a reason for these two peaks, so that essentially both things are correct.
yes, so actually that is a good point. So I'm not excluding that there are ice-like or water-like. There may well be ice-like and water-like structures.
Because otherwise it won't be so far.
But there's not exactly, but there's clearly not a sort of two-state model. There's a continuous distribution of states. And so that's what the comment I was trying to make. I can't tell how heterogeneous or homogeneous it is. The thing I can say that there are no two distinct substates. Yeah.
Okay, I think it's time to thank Misha once more and